Hello students, welcome to the new lecture, and in this lecture we are going to discuss from the activity 3 and so on. Okay, so first of all, if you have the good internet connection, watch this lecture at 480 pixel. Okay, why? Because at 480 pixel the lecture will be good, and if you have the slow internet connection you can watch at 360 pixel. Okay, so first we'll start with the activity 3. In the activity 3 we have to do the observation of the seeds of the pea or bean pod. So what is meant by bean pod? Okay, so let us consider the bean pod will be in this way. Okay, the bean pod will be in this way. In here the seeds will be there. In there the seeds will be there in this way. So we have to observe the seeds and also pod of it, how the shape of the pod and how the shape of the seeds and so on. So we will get some ideas about it. Okay, we are going to see about it more in the coming lecture. Okay, so there are a few questions. The first question is, can you find two similar seeds there? Okay, actually you don't find similar seeds. Why? Because based upon the genes or traits which will be passed into the seeds, they may be different. Okay, there may be different or may not be similar. Okay, what makes them vary means different. How? Why the one is different with the other? So why? Because the different factors or genes. Okay, different factors or genes makes them vary each other. Okay, or vary from each other. So this seed may be different. Okay, and this seed may be different, this is different, and this is different based upon the genes or factors which they obtain. They are different from each other. Okay, so the next question is, why variations are important, how variations are useful for organism or population. So see here, the variation means the changes. So the changes that occur in the organism, whether it may be genetic. Okay, the changes may be genetic. Okay, or evolutionary. Okay, or not evolutionary, the environment. Okay, the weather may be to the genetic or evolution or environment. So these changes will occur. These are nothing but variations. So these variations, what they are going to do, these variations are helping the organism to survive. Okay, so these variations are helping the organism to survive. So as a result, this organism can survive better in the nature. Okay, that is first advantage. And next advantage is the species will be formed, new species will be formed due to the accumulation of the variations. So next, the variations, one variation, two, three, four, and so on. There are a number of variations are occurring or accumulating in the particular organism. So in that case, if there are many variations accumulating in the particular organism, this leads to the formation of the new species. Okay, so as a result, new species are formed due to the accumulation of the variations. Okay, so yesterday we have discussed how the person will become how the person turns into darker skin when he is living in the sunlight, very huge sunlight areas. Okay, that is due to the changes in his skin. Okay, more sunlight making his skin to become a darker color. Okay, so that is due to the environment, and one is the genetic. Okay, these two are important. So next is Mendel. Okay, the Mendel and his experiment. So what is his experiment? Who is Mendel first? So first, the Mendel, he is a monk. Okay, Austrian monk. So he is a Austrian monk, and he belongs to the 19th century, and he provided the detailed experiment of the passing variation from one generation to another generation. Just now we have discussed what are variations means, the changes. Okay, the changes that are occurring in the parent, will be passed to the children. Okay, that will be passed to the children. And we, in those days, they did not know how these changes are passing from parents to the children. So this Mendel, so the scientist, he proved how these changes will be passed, how these characters are passed from the parents to the children. Okay, so for that he did a detailed experiment, so like monohybrid, dihybrid, and so on. So these all are the experiments done by the Mendel, and he said that how the variations are passing from one generation to the another generation. So as a result, he provided valuable information for us. So as a result, we consider him as the father of the genetics. Okay, why? Because he is the main person to introduce the genetics for us. Okay, and performed his experiment on the pea plant in his monastery. So monastery means it's a place or building. It's a place or building where the monks live. Okay, monks means they dedicate their life for the god. Okay, so in his monastery, or the place where that these monks are living, so that is known as monastery. In the monastery there is a garden. Okay, it means a place where the different plants were grown, that is nothing but garden. So he did such kind of experiment for 7 years. So 7 years is not a small thing, so it is a huge time. So he has taken huge time so for the performing of these important detailed experiments. Okay, so it takes a lot of patience, lot of care, and lot of effort. Okay, so because of these all hard work, we will call him as the father of the genetics. Okay, and next, a detailed experimental procedure and conclusions were presented in his research paper. Whatever the work he is done, whatever the work he is done, he presented and he wrote in his research paper and published. So publish is nothing but we can see or we can read and so on. So that is nothing but publishing a research paper. Okay, so next, there is another question. What made the Mendel to select the pea plant only? There are many plants. So tomato plant, papaya plant, and apple plants. There are many plants. So leaving all those plants behind and leaving all those animals behind, why he selected only the pea plant? What is the reason for selecting only pea plant? So there are few main important factors. So he carefully observed the plants. He observed many plants like potato, tomato, and so on, different plants. Among them, pea plant is also one. So after careful observations in all these plants, he came to conclusion that these pea plants has some advantages. So what are these advantages that is present in the pea plant? So first one is well-defined characters. Okay, it has some characteristics like stem length, flower color, flower height, and also position of the flower, and so on. These all are the well-defined characters. He did not found such kind of characters in the other plants. So next is the bisexual. What is meant by bisexual? So the flower, let us take this is the flower. Okay, the flower contains male reproductive organ and also the female reproductive organ. So these are stamens and anthers. So the male reproductive organs are there, female reproductive organs are there in the particular flower. In that case, we will call it as a bisexual. Bi means two, sexual means in this term it refers to male and female reproductive organs in the particular flower. Okay, so next is the predominantly self-pollinating. It means the first of all this pea plant try to have self-pollination. What is self-pollination? Let us consider this is the flower. Okay, and in the flower this is the female reproductive organ and this is the male reproductive organ. Okay, stamens and also pistil. So okay, so the pollen grains are going to fall on it. The pollen grains first fall on it on the stigma. So this is the anther, this is the stigma. So first of all the flower try to have self-pollination. Self-pollination means the transfer of pollen grains from the anther to the stigma of the same flower. Okay, this is known as self-pollination. So next, what is cross-pollination? Let us consider this is the flower and this is the female reproductive organ, this is the male reproductive organ. Okay, so what happens? So the pollen grains first will not fall on the same stigma. Okay, so this from this anther the pollen grains will go and fall on some other flower. Okay, so it means the transfer of pollen grains from one flower to the another flower. Okay, so that is nothing but cross pollination. Cross means in between these two flowers. So between these two flowers, that is nothing but cross pollination. So within the plant, the transfer of pollen grains within the same flower, that is known as self pollination. Self means within, cross means between. Okay, so next is suitable for cross pollination. So if we are interested, or if you are interested, you can do the self pollination of a flower, and if you are interested to do the cross pollination, you can easily operate these plants, or you can easily do such kind of self cross pollination also. Okay, it means suitable. It is very easy to do the cross pollination in these flowers. Okay, next it is an annual plant. So what is meant by annual? Annual means within a year. Within a year, its life cycle will be completed. Okay, this is about the main important reasons for selecting the pea plant by the Mendel. Okay, next the Mendel worked on nearly 10,000 pea plants. So 10,000 is not a small number, it's a huge plants. Okay, in these 10,000 plants there are varieties. Okay, how many varieties? 34 varieties are there in these 10,000 pea plants. Okay, why he did such kind of huge experiments on huge number of plants? So just to find out answers. So first, just to find out the answers for his questions. So this is questions. Okay. So to find the answers for his questions and his doubts, so he did such kind of experiments, and such kind of information helped us a lot to understand the genetics. Okay. So after careful observation, the pea plants differ from one another. Okay, one another in many ways. Example: some plants were tall, some plants were dwarf, some are round, some are wrinkled seeds, and so on. It means these all are the different variations that can be seen in the pea plants. If you observe, some pea plants were smaller. Okay, some pea plants were taller. So why such kind of variation? This is due to the characteristics or traits that is present in the plants. Okay, so hence Mendel chosen 7 pair of contrasting characteristics. Okay, here we have seen a term that is known as seed coat. What is seed coat? Seed coat means a layer, okay, microscopic or thin layer around the seed. Such kind of thin layer around the seed, that is known as a seed coat. Okay, so for example, if you see a groundnut, on the groundnut you can take out a thin layer around it. Okay, that is seed coat. Okay, so just now we have seen Mendel chosen some important characteristics. So here mainly in that he has chosen 7 pairs of the contrasting characteristics. Okay, in this table you can see what are these 7 characteristics. So first character is the color of the flower. So color of the flower may be white or purple. Okay, white color or purple color, this is one of the contrasting characteristics. Next, the position of the flower, where the flower is present. Let us consider this is a plant. Okay, let us consider this is a plant, and the way the flower is present, whether the flower is present on the top or in the middle or at the bottom. So this is about the position of the flower. In this case, the flower is mainly seen in two, that is axial or terminal. Axial means what? So axial means in between. Here in this case the flower is present in between. So terminal means on the top. So here the flower is present. So this is the axial and terminal position of the flower. Okay, so next, the color of the seed. So after the formation of the seed may be two colors. So that is whether it may be yellow or whether it may be green color. Okay, we will discuss about the formation of yellow seeds and green color seeds in the coming lectures. Okay, next is the shape of the seed. The seeds may be round and smooth, and the shape of the seeds may be wrinkled. Okay, the seed may look in this way, seed may look in this way, or seed may look in this way. So this is completely due to the genes or characteristics that are characteristics passed to the seeds. Okay, so next, the shape of the pod. So this is the pod. Okay, so this is the pod. If you see the pea plant pod, so in the market they will sell such kind of pea pods. Okay, so here in this the seeds are present. In this the seeds are present, and see how the structure of the pod is. Whether the pod is wrinkled in this way, okay, wrinkled in this way, or the pod is super shiny or full, or whether the pod is not full. Okay, so observe such kind of characteristics. Okay, this is one of the reason for selecting the pea plant. So next is the length of the stem, how much length it is. Okay, some plants, let us consider this is the ground. On the ground, if you observe, the plant may be this much height. Okay, the plant may be this much height, and the another plant may be this much height. Okay, this is the pea plants only. Okay, why there is a difference in the height? Okay, this is due to the characteristics or traits. So this is also one of the main characteristics to select the pea plant by the Mendel. Okay, so here the pods also different colors, either yellow or green in color. Okay, so next there is one question, that is how do parent plants pass their traits to the seeds? So traits is nothing but characteristics. So during the sexual reproduction, so sexual reproduction means so where the fusion of the gametes like spermatozoa and also ovum. Okay, so here in the sexual reproduction, the factors or traits will be passed to the gametes. So just now we have discussed that is spermatozoa and ovum in case of animals, but in case of plants, pollen grain and also ovum. Okay, so gametes fuse to form zygote. So after fusion of the gametes, the zygote will form, and eventually the zygote will develop into an organism, or in case of plants the zygote will develop into a seed. Okay, a seed. Okay, so it means the factors or characteristics will be passed from a cell to the gametes, from the gametes to the zygote, and zygote to the seed, and the seed will be grown into a plant, and these plants will show such kind of characteristics that has been taken from the plants or parental plant. Okay, next there is small little bit information about the pea plant. So the peas were the pea plant or peas were present in many parts of the world. So especially Afghanistan in 2000 BC, and Harappa, Pakistan, and Northwest India in 2200 BC to 1750 BC. Okay, and in case of Gangetic Basin and also southern India, it is around second millennium. So there is a question: what is meant by millennium? Millennium means thousand. Okay, millennium means 1,000 years. Okay, that is known as millennium. So the pea is an annual and also cool season plant. It means within a year its life cycle will be completed, and it is a cold season plant, means it will grow in the cold areas, okay, where there is a less temperature. So it contains different kinds of nutrients like vitamins and minerals. So especially vitamins like A, D, E, C, and K. So those are vitamins, but here if you carefully observe, only vitamin D is missing, and except vitamin D all vitamins are present, and minerals like calcium, iron, magnesium, manganese, phosphorus, sulfur, and zinc. These all are the minerals that are present. So these minerals and vitamins, these both are important for the normal body function. Okay. So next is Mendel hypothesis. So what the Mendel is thinking about his observations. So based on his experiment, he observed that the characters carried as a trait. So in the last lessons also we have discussed what is meant by trait means these are the characteristics that can be observed, okay, are a pair of factors. Okay, pair means what? Pair means two. Okay, so pair means two. Example, pair of shoes means what? A right shoe and also left shoe is known as a pair of shoe. Okay, so these traits or characteristics are going to pass to the children or the pass to the pea plant seeds, so in the form of two. Okay, that is nothing but a pair of factors. So distinguished traits of the same characters were present in the population of an organism. Population means they are closely related. So in the closely related plants or organisms, these traits are same, almost same, or same characteristics. Okay, the traits shown by the pea plant. The traits shown by the pea plant must be in the seed which are produced by their parental plant. For example, so a seed, okay, a seed has a yellow color and another seed has a green color, okay, the small y. Okay, so they are going to pass this information to the seeds, okay, that is nothing but is a progeny. Okay, and the whatever the characteristic that you see in this seed will be coming from the parents. Okay, so this is about this sentence. Okay, so next is the procedure followed by the Mendel. What procedure he followed for his experiment? So the first one is Mendel selected varieties of the plants. So varieties means different characteristics. Different in the plants there are different characteristics. That is nothing but varieties of the plant. So based upon the seeds, and he grew more plants out of them. So he took different varieties of the plants, he collected the seeds, and he grew more, and he produced many plants or many seeds. Okay, self-pollination allowed in a plant in a particular way. First of all, whatever the seeds he obtained, he did self-pollination. Means what is self-pollination? Means the pollination within the flower. Okay, within the same flower, that is nothing but self-pollination. So as a result, by doing the only the self-pollination, he procured or produced the pure varieties. Remember, this is important. How do you produce the pure varieties? So pure varieties of a particular plant can be produced by the self-pollination only. Okay, just now we have discussed what is self-pollination. So transfer of pollen grains from anther to stigma of a same flower, that is known as self-pollination. So through this self-pollination, he produced the pure varieties. So what is meant by pure varieties? Example, just now we have discussed the traits or a pair of characteristics. So let us consider this is a pair and this is a pair. So yellow colored genes. So here if you see, both are the a pair. Okay, this is one and this is one, so together we are calling it as a pair. So a pair of the genes, so both are same, so this is pure. For example, let us take another example. This is y, so this is small y. So here it codes this gene is responsible for the yellow. Okay, so and this gene responsible for the green. Okay, are both same? No, both are not same. So in this case it is not pure. In this case it is pure. Why? Because this is for the yellow and this is for the yellow. So both are same. So by the self pollination, by the process of self pollination, you can produce such kind of pure varieties. So you cannot produce such kind of pure varieties with cross pollination. So cross pollination is not suitable for the production of the pure varieties. So Mendel followed the same procedure. He did self pollination of a plants to produce such kind of pure varieties after over a generations. Okay, repeated self pollination means generation after generation, generation after the generation, only self pollination were followed. So pure varieties were allowed to grow and flowering, and then these plants were cross pollinated after getting of the such kind of pure. Okay, let's know this is pure and this is also pure, and after getting such kind of pure they were cross pollinated. So cross pollination means the transfer of pollen grains from the anther of the one flower to the stigma of the another flower. So the cross pollination will be done in this way. Okay, and next is monohybrid cross. So here if you see the word, this is a monohybrid. What is monohybrid? Mono means one. Okay, here the mono means one. Okay, hybrid means only one character or contrasting characters. So here in this case color. Okay, in this case color, we are going to focus only on the color only. What are the colors? The colors may be yellow. Okay, the colors will be yellow and another color will be green. Okay, we are going to focus only on the color, not the size of the seed or shape of the seed. So remember, we are going to focus only on the color. So this is only one character, hence that is nothing but monohybrid. So what is meant by hybrid? Hybrid means two characteristics. Let us consider this is one and this is another one. Okay, so can you see two different things are present in it? So this is known as hybrid. Okay, this is known as hybrid. It means characteristics of two different things. Okay, so in monohybrid cross, the cross pollination is made. So up to now self pollination is there to obtain the pure varieties. Now we have to see how the characters will be passed to the children or to the young progeny. So that is to observe that we have to do the cross pollination of the plants. So that is between the pure yellow and pure yellow seed bearing and also plants of the pure green. So here in this case the green represents the capital Y. Okay, capital Y represents the pure yellow. Okay, or yellow color. So the green color, for the green color we are using the small y. Okay, here how these are obtained by the pure, these are the pure, how they are obtained from the self pollination. From the self pollination they are obtained. Okay, self pollination, so pure will be obtained. So now we have to see how these characteristics are going to pass to the progeny. So first, if you do the cross pollination, so you will get F1 generation. Okay, what is meant by F1? F1 means filial. Filial means son. Okay, so or you can call it as a progeny. So F1, all plants produce were yellow seeds. So whatever after doing the cross pollination, between these two pures, you will get F1 generation. In the F1 generation, so the color all are yellow only, yellow 100%. Okay, yellow 100% or 100%. Why? Because so here one is dominant, another is recessive. We will discuss about the dominant and recessive in the upcoming lectures. And now see, in the F1 generation all were yellow only. Okay, So here this is the green, this is the yellow, but here in the all seeds you can see yellow color only. Okay, and next is second filial. So how you will get second generation or F2? You will get F2 again the self pollination. Here you are going to do the self pollination. By doing the self pollination, here you will get the F2 generations. So in the F2 generations the color is different. How? Which color? Yellow. Okay, yellow will be there and also green will be there. Okay, so this is about the color. So yellow will be 75 percentage, yellow will be 75 percentage, and green seeds this is 25. Okay, not 20, the 25 percentage, 25% green seeds will be seen. So this is about 3:1%. So let us consider the four seeds are present. Four seeds are present or produced. In the four seeds, the three seeds will be yellow and one seed will be green. So hence you can write in this way, 3:1 ratio. 3 refers to the yellow and 1 refers to the green. Okay, and Mendel observations in the monohybrid cross. So what he observed in this monohybrid cross? So he observed that F2, okay, or a set in a set of F2 generations. So where the homozygous dominant is 25 percentage. Homozygous means what? Homo means same. Okay, in this case, capital Y is homo. Why? Because it both out of these two genes, both are for the yellow color. So in this case we can call it as a homo. So what is hetero? Hetero means one should be one type, another should be another type. So in this case capital Y and small y both are different. One codes for the yellow and one codes for the green. So in this case they are different. So this is hetero, okay, and this is homo. Okay, so next, in the second sentence, we are discussing that a set of P's in F2 generations were heterozygous yellow about 50 percentage, which produce yellow-green seeds in 3:1 ratio. And in the F2, a set of F2 generations where homozygous ratio is about 25%. This is not 20, 25%. Okay. So 25 percentage and keeps the producing green seeds on self-pollination. Okay. So in this we will do one small activity or small experiment. So this is observation, that this is known as checkerboard. Okay, checkerboard or Punnett square. So by this way we can get information how the progeny will be. So these are the gametes. So these are the parents. These are the parents. So remember this is about the gametes. So capital Y produces what? Capital Y only. Okay, capital Y produces capital Y small y. So capital Y small y, it means small y and capital Y. So small y produces small y. So generally in the genetics we have to write the capital letter first, and after the capital letter you have to write the small letter. But in the test tube in this way is given, so it is small mistake. Okay, so in the textbook the small y and capital Y is given, but you should not write in this way. You should write first capital and next small. So as we have shown in this way. Okay, so here if you see what they are saying that 25 percentage produce yellow seeds. So if the capital Y is present, so it will be yellow. Why it is will be yellow? Both are dominant, as well as these capitals will be expressed, so the seed will be yellow. Okay, in this case the next will be yellow only, capital Y small y, next small y capital Y, both are yellow only. Why these are yellow? If you see in these two, in this set, okay, out of these two, one will be capital Y. One capital Y means what? Capital Y refers to the yellow. Okay, so this will be yellow. Next is also yellow. Why? Because in these two genes, one gene will be dominant, one gene is dominant. So due to the presence of dominant genes, the seeds will become yellow, and here the green is recessive. Small y refers to the recessive. It means it will not be expressed. So only the yellow color will be produced in the seeds. Next is small y. So the small y. So what is the small y indicates? Do you find any capital Y? No. If the capital Y is not present, dominant gene is not present. So as a result, the residue will be expressed. So both are residue. As a result, the residue will be produced. So this is nothing but green. Okay. So take the ratios. 25 percentage. Okay. So 25% is homozygous. Okay, it means same genes. And 50%, 50%, the 50% will be heterozygous, and 20% or 25 percentage is homozygous. So this is homozygous dominant, and this is homozygous recessive. Okay, so understood about the homozygous. Here you can see, okay, here you can see homozygous recessive. Okay, so this is about homozygous recessive. And here the first, in the first sentence, homozygous dominant 25%. Can you see homozygous dominant over here? Yes. And 50%, okay, the 50% is heterozygous yellow. Okay, so can you see the heterozygous yellow? 50% heterozygous. Why? Because one capital and one small y. Okay, so this is about Punnett square. By you doing this, you will come to know about the progeny. Okay, and Mendel assumptions. What is assumption means? Assumptions what the Mendel is thinking. So Mendel is thinking that so every pea plant has a particular character determining agents. So in this case, we came to know that these determining agents are nothing but genes, that is capital Y, small y, and so on. So they are responsible for the color. Okay, they are responsible for the color. Here we are calling it as a trait. Okay, trait called a factor. So green color genes factor and yellow color factors. So each plant has the two factors. So what are those two factors? Yellow and green. These two are the factors. So it is known as a pure. So if they have the same, that is known as pure. How what will be pure? So capital Y, both of them has that yellow colored genes, no green color, hence they are known as pure. How the pure is obtained? Just now we have discussed. So self-pollination of the individual or parental plants will produce the pure. Okay, the self-pollination. So here the capital Y is pure, or the small y is also pure. Why? Because here you can see, so only green color genes, okay, green color genes, or no other genes are present, so it is also pure. How the pure is produced? Again same, the self pollination, from the self pollination. Okay, so in if the plant has, yeah, this is thing, if two factors are same in the plant, it is known as a pure breed. Okay, so example, capital Y, it is a homozygous, or small y is a homozygous. If a plant has two different factors combined, it is not pure. For example, capital Y, small y. So capital Y, small y. So are they same? No. One gene is for the yellow. Okay, one gene is for the green. So in this case, they are not same. Hence, it is known as heterozygous. Okay, and they are not pure. So how this will be obtained? They will be obtained by the cross pollination. So they are obtained by the cross pollination. If the cross pollination is available, in this case such kind of genes will be formed. So next is assumption 2. In the assumption 2, the Mendel is thinking that during reproduction, one factor from each parent is taken to form a factor, or to form two factors or a pair in the progeny. Let us consider this is a capital, this is one parent, and this is another parent. So this is a cross pollination. So as a result, one gene come over here and forms capital Y, another gene comes from here and forms a small y. So this is during the cross pollination, or this is due to the sexual reproduction. So sexual reproduction means the formation of the gametes. Gametes will be formed here. The yellow color in the gametes, the yellow color gene is available, and then another gamete which is coming from the another parent, green color genes are present. So this is how the genes will be passed, or the traits will be passed from the parents to progeny. So next is, and here you can see this is a pair. What is meant by pair? Pair means two. So this is one and this is the second. So two are present. So in the assumption 3, if the factors are mixed, one will be dominant, another will be get expressed. So if you see, and if you see here, so one is capital, okay, one is capital and one is small. Always the capital refers to the dominant. One is capital, it refers to dominant, and second is the recessive. It will not express, it will act as a recessive. So what is meant by recessive? It is suppressed by this dominant gene, that is capital Y. Okay, and here in this case, example capital Y small y. Here the Y capital Y is dominant and small y is recessive. So if such kind of genes are available, so the seed will be yellow color only. Why? Because the capital Y is there, it is dominant. So based on above examples, all seeds of F1 generations are yellow due to the presence of dominant genes or factor expression. Here is another Punnett square or a checkerboard. You can do the you can see the results. So here the capital Y and also small y. So here capital Y, small y, and next is capital Y, small y, and capital I and small y. So can you see how many green seeds are there, how many yellow seeds are there? So first one will be capital Y, small y. So let us take the second one, capital Y, small y, and here also capital Y, small y, and here also capital Y, small y. Can you find dominant genes in each and everything? Yes, capital Y is the dominant. It is present in each and every seed. So if the capital or dominant gene is present in each and every seed, so all seeds were yellow, or you can say 100% yellow seeds. Okay, and this is about Mendel and his assumptions and so on. So I hope this lecture is helpful to you, and in the next lecture we are going to discuss about the phenotype, genotype, and the self-pollination in F1 and self-pollination in F2 and so on. Okay, and we will discuss the next lectures, all these lectures in the next lectures. Okay, if you have any doubts or questions, you can post it in the comment section. Okay, in the comment section you can post it. And if you have the slow internet connection, watch at 360 pixel, and if you have a good connection, internet connection, you can watch at 480 pixel or above 480 pixel. Okay, and post your comments or questions or suggestions in the comment section so that we can reply to it and so on. Okay.